I would like to introduce to you Dr. Paula Rozo-Lopez. She is a biological researcher working on insect virus interactions, including arboviruses and insect-specific viruses. She has a broad range of field lab and computational experience working within various insect systems. Her current research as an NSF postdoctoral fellow at UT aims to understand the evolutionary and transmission patterns mediating the heritability of insect-specific viruses in agriculturally important insects. So with that, I'm going to turn it over to Dr. Rosa Lopez.
Oh, can everybody see this okay?
Yes, perfect.
Alrighty, so thank you everybody for joining in. I'm very happy to be here with you, and I'm just going to talk to you a little bit of the research we do with insect viruses and try to show you a perspective of how this affects disease and food security. So just to start with a quick introduction, you guys might know that insects are one of the most abundant and diverse groups of animals. There are around 1.5 million species described, which accounts for more than half of all described species of living organisms in the world. So insects are found nearly in every terrestrial habitat and also in a wide range of aquatic environments. And as all living organisms, insects have evolved around microbial infections. But it's not only until the recent decades where sequencing technologies have allowed us to discover a remarkable diversity of DNA and RNA viruses. So when we're talking about the insect virus composition, which we call virome, we can find insect-specific viruses, which, as their name suggests, they're viruses that only infect insects. But we can also find vector-borne viruses, which are viruses that exist in transmission cycles between insects that we call vectors and either plants or animals that we will call host. So insects and their viral infections play viral roles in ecosystems. For example, they act as pollinators of crops. They can also act as food sources for other species, or even helping with the decomposition and nutrient cycling. However, some of these insect virus interactions can also have negative impacts by acting either as pests of crops, for example, of vectors of human and animal disease. So when we're specifically looking at the negative impacts of insect viruses on human health, we see that in the last decade, human populations are becoming significantly more exposed to these viruses. For example, the last year here in the United States, we see diseases such as West Nile virus and dengue virus to had a dramatic increase in transmission. In the case of West Nile viruses, last year only presented around 1,800 human cases with active transmission in 48 states. Whereas for dengue virus, there were more than 10,000 infections reported, but states like Florida and California have very well-established local transmission cycles. So looking beyond the human cases, the economic burden of this disease is also very significant. For example, West Nile virus has an estimated cost of $800 million since it first appeared in 1999 here in the United States. Whereas for dengue, we see that the active transmissions across the entire continent has a calculated burden of $2.1 billion annually. In the case of food security, insect viruses also have a direct impact in agriculture. So for example, some insect pest and plant viruses transmitted by these insects can have a devastating impact on crops. While the precise amount of money spent by farmers to control these two interactions is very difficult to pinpoint, studies on specific crops indicate that, for example, corn and soybean farmers may spend something between $17 to $26 per acre, while high-value crops like, for example, potatoes can have costs exceeding the $200 per acre. But there is also viruses vectored by insects that affect animal health and therefore agriculture. So for example, blue thumb virus, which is directly affect agriculture in the United States with a calculated impact in between the insect pest and the viruses can exceed $100 billion annually. But not all is bad when we're talking about insect-virus interactions. There is also a positive side of the insects and their viruses. For example, insect-specific viruses can infect and kill only very specific species of insects, which allow us to use them as biocontrol agents for agricultural pests. On the other hand, some insect viruses have shown to interact directly with the insect vectors and the pathogens they transmit, which can help us to use them as a mechanism to lower disease transmission. So in that order, the research we do here at the university help us to understand what mechanisms drive changes in insect populations, virus diversity, and pathogen transmission. And when we link humans, animals, and plants and the environment, we actually can address the full spectrum of disease control. We can look from prevention to detection, preparedness to response, and even management. So what I'm aiming today for this talk is to show you some quick examples of how we can approach the understanding of insect-virus interactions in the laboratory. Specifically, I'm going to show you some examples of my research where I have used a wide range of insects, multiple viruses, and a combination of techniques to better understand the interaction between insects, the viruses, and how they can modulate animal and plant disease. So one of the insects I have worked with are Culicoides biting midges. Culicoides are very small flies, as you see here in the picture, where those lines show the size of millimeters. Midges are often found near water sources and vegetation, and they're very well known among farmers because of the irritating and painful bites. While they're primarily nuisance, they can also transmit several viruses that cause disease to animals. And because that close association with livestock, they are considered a very important agricultural pest in farm settings. So when we're talking about virus transmission by midges, we can summarize these interactions in 4 steps. The first step is going to occur when the adult females need to feed on blood of an animal to get enough nutrients to produce eggs. And during this blood feeding process, they can ingest viruses present in the blood of sick animals. Then, while that blood meal is being digested, those virus particles inside the midge stomach are going to infect some cells. Then they're going to replicate, so make more copies of their own and produce more virions that then will disseminate throughout the insect body. And after that dissemination, those viruses will reach and infect salivary glands, which are located below the head, so they can finally be released in the saliva when the midges feed on the blood of a new host. So they need to feed multiple times in a night cycle to produce multiple egg batches, and this allows either the acquisition of the virus or the transmission of the virus that was already acquired. The duration of this cycle in between picking up the virus infection and being able to transmit it is temperature dependent, but it generally ranges between 4 to 12 days, and it mainly depends on the combination of the virus species and the vector species. So knowing the basics of vector-borne transmission, one of the first questions I had in mind was why culicoides are such a good vectors and how they contribute to the transmission of viruses affecting animal health. So a simple first approach was to understand how the volume of ingested blood might affect virus transmission. And this is important because culicoides, as I mentioned before, they're very, very small insects. So in comparison to mosquitoes, which you see here in these pictures that I have in proportion, it is expected that they will ingest very little blood. So the larger the volume of blood ingested, the bigger chance of ingesting more virus particles. So in theory, larger insects can serve as better vectors of disease. So to understand the transmission potential of a smaller vector such as culicoides, what we did first was calculating the amount of blood that they can ingest. And this resulted in a volume of 100 to 150 nanoliters, which when we put in perspective, this is about 3% the volume of blood you will find in a drop of blood here, like when you prick your hand to take a glucose test, for example. Then knowing this volume, we wanted to understand how the virus concentration in the blood meals affect this virus transmission. So we use a system to deliver artificial blood meals that you see here in this picture that allow us to serially dilute several blood meals with different infectious doses. Then we fed the insects, and we selected only the insects that were fully fed on blood. And as you see here, besides the small size, it's very obvious which insects were successfully fed. Then we held these insects for 10 days, and we test head separating from the bodies as an approach to evaluate a transmission potential. So remember, those heads are going to have the salivary glands attached below them. And when the virus reaches that salivary gland infection is when it's ready to be transmitted in the saliva. So we know that those positive heads will mean that those midges can transmit the virus if they feed on the blood of a healthy animal. So what we found is that indeed the oral dose shapes the transmission potential. So what you see in this graphic here is in the x-axis, you're gonna see the different amount of blood delivered in the blood meal. So the different concentration of virus particles that we put in each blood meal. And in the y-axis, you're gonna see the percentage of virus positive, either bodies that is depicted in black or heads that are depicted in orange. And we see here that with higher infectious doses, there is a higher percentage of positive tissues. However, only doses above the 5.2 range resulted in disseminated infections in the head, which indicated those insects that are going to have the transmission potential. So further analysis allow us to calculate that 6.3 was the minimal oral infection rate required to disseminate in 50% of a population of midges that fed the repletion on an infected animal. And what are all these numbers really telling us? So is that besides ingesting very small volumes of blood, as you remember, it was between 100 to 150 nanoliters, when we put this 6.3 value in the context of the disease epidemiology, where we see animal titers, so the amount of viruses that we can find in infected animals are going to range between 7 and 9. So we see here with our number that midges that fed on an infected animal can easily develop enough disseminated infections with transmission potential to significantly contribute to the virus epidemic. So another example of where research can allow us to understand virus transmission involves also putting the results in the context of blood feeding behavior. So as I mentioned before, midges and many other vectors that feed on blood will feed multiple times in their lifetimes to sustain multiple egg-laying cycles. So in the case that the vector acquires the virus in the first blood meal, we really need to understand what is going to happen with the transmission potential after they feed multiple times on healthy hosts. So what happens when they feed for 2 and maybe 3 times more after they ingested the blood meal? So to do this, we again use our artificial delivery system to evaluate the effects of ingesting 1 and 2 additional non-infectious blood meal. So we infect the midges with the virus in the first meal, and we provide them additional blood that had no virus at all to see what was the impact of these feedings. So what we see here in this graphic is we observe the virus amounts in the y-axis. So the higher the dots go, the higher amount of virus we detected in those midges. And we compare that to the midges that fed multiple times that you see in the gray dots versus the ones that fed only once, that are the black dots. And we see here a significantly higher amount of virus in the groups that feed multiple times, but also the percentage of midges that we detected as positive after multiple meals also increased, being from 50 to 60% in the groups that fed only once, to increasing from 79 to 93% of the midges with additional blood meals. So what these results suggest is that feeding additional times on healthy animals increases the likelihood of virus transmission by infected vectors. But also, if we account this natural behavior, feeding multiple times, we've seen that the more successful a vector is from feeding multiple times and producing more egg-laying cycles, the higher the chance is going to be that in that process, that vector is going to be able to transmit better and more viruses to the infected animals. So another perspective or a different approach to understand virus interactions can involve the virus itself. So for example, there are some characteristics in the virus genetics that can contribute to enhanced transmission during epidemics. And this is the case of a vesicular stomatitis virus, also known as VSV. This virus is transmitted by culicoides midges, and it can cause disease in cattle, pigs, and horses, but can also infect humans. The virus clinically presents in the animals by showing these vesicular lesions that you see here in the mouth, sometimes in teeth and hooves, and can lead to secondary bacterial infections. Although this disease results on its own, all the infected animals are immediately put in quarantine. There are also trade embargoes for animals that had VSV, and the production, for example, in cattle decreases significantly. So it generates a pretty high impact, economic impact to all those infected ranches. So this virus is usually endemic in Central and South America, where it occurs as an annual outbreaks. But here in the United States, VSV presents with epidemic outbreaks every 5 to 10 years. So an interesting example of this transmission dynamics was observed by some of our collaborators in 2012 when they detected two closely related viruses circulated endemically in Mexico. So they named them 1.1 and 1.2. Surprisingly, the one named 1.1 is spread north, causing a severe outbreak in the southwestern states, whereas the closest relative, what they name 1.2, stayed circulated in its endemic area in Mexico, but never left the endemic area. So to understand what was different between the endemic and the epidemic viruses, our group of collaborators in USDA Plum Island sequenced the virus and found that there are only 7 amino acid substitutions with significantly protein effects. So only 7 amino acids have the significant effects between the endemic and the epidemic strain. Then they experimentally infected the pigs, as you see here, the lesions in the snouts, and they observed that the pigs infected with the epidemic viruses, so with those viruses that escaped the endemic area and caused the outbreak here in the United States, had bigger lesions, but also have a prolonged periods of fever, higher virus titers than the animals that were infected with endemic strain. So we see a higher virulence by these epidemic lineages. So when I look at the differences in both viruses' ability to infect the vectors, what we're observed first is here in the graph in the left is that the percentage of virus-positive bodies and the titer, so the amount of virus we found in the vectors, were significantly higher in those vectors that fed on blood infected with the epidemic lineages, so the lineages that caused the disease outbreak in the United States. But I also saw that at least the percentage of heads, so those midges that had disseminated infections for transmission potential, was also higher in those vectors that fed with the epidemic viruses. So what we see here is that in addition of affecting virulence in sick animals, these small genetic changes in the virus has significant effects on the vectors. So we see that epidemic viruses have an increased transmission potential, which allows them to be more successfully to spread outside the endemic area. And that's why we believe they're causing outbreaks in the United States. But They might last only a single year, so stopping in the winter when all the vectors die out. But very, very often, we see that the same virus lineage reemerge in late spring for a second year outbreak. So in this case, there has to be a mechanism in which the virus persists in the absence of infected animals. So a big question and a big part of my doctoral research consisted in exploring alternative mechanisms that can help us to explain this virus persistent in vector populations without blood feeding. One of these alternative mechanisms is venereal transmission, in which the virus is transmitted between the female and the male vectors through copulation. This is a unique process that accounts not only for the that occurs only in the viruses that rapidly disseminate in the vector and infect multiple tissues, including the reproductive organs, as you see here, but all the pink areas in the pictures below. So in order to test if this venereal transmission was even possible with midges, the first thing we did was taking newly emerged females and males to make sure that they haven't copulated before. We placed them in a cage and offered them a high titer blood meal containing VSV. So generally, these male midges will copulate with females where they're feeding on blood. As you see here in the picture, the male grabs the female with the claspers and they attach together, and that's how they copulate. So after this oral infection, we sort those males, we sort the midges, and we offer them additional null infection blood meals every 4 times with and putting new males in the cages to stimulate that copulations through multiple egg-laying events. So what we see here in the graph in the x-axis is that we tested those males that copulated with the females, for the first egg-laying event that we named 1GC, the second and the third. And what we see here is that those midges, those males that copulated with orally infected females after the second and the third egg-laying event tested positive for the virus. So 76% at the end of the second egg-laying event, 15% at the end of the second egg-laying event, and 76% at the end of the third egg-laying event. So what these results indicate is that females that acquire the virus by feeding on the blood of an infected animal can pass the virus to uninfected males through copulation. But if you remember, culicoides males cannot transmit the virus to animals because they don't feed on blood. So in order for this transmission to be relevant, those infected males need to be able to pass the virus to uninfected females. So those uninfected females can restart the virus outbreak when they feed on blood. So to test this transmission from venereally infected males, so those males that acquire the virus after copulating with orally infected females, we put those males with uninfected females, offer them a blood meal with no virus at all, just to stimulate the copulation. And what we saw is that around 9.5% of those females tested positive for the virus after copulated with venereally infected males. So what our results indicate is that those males that acquire the virus by copulation can easily infect transfer the virus to uninfected females through mating. And lastly, to explore how relevant venereal transmission it is as a maintenance mechanism, that to see if it will actually allow for the reemergence of the virus, we tested whether those venereally infected males can transmit the virus by blood feeding. So as we did before, we tested the body separate from the heads, and remember that those heads have the salivary glands. So if we find them positive, they will have potential transmission. And what we find here is that actually, and very surprisingly, 10% of those female heads tested positive for the virus. So what our results indicate is that venereal transmission can indeed potentially result in transmission via blood feeding. So it is possible for vesicular stomatitis virus to overwinter in vector populations. For example, during those mild winters when the vectors survive without blood feeding. And then the virus will reemerge when the conditions are ideal for vector-borne transmission again. So when the spring comes and those females need to feed on the blood of an animal to lay eggs, some of those females are gonna be positive and they can restart the outbreak for the second year. So what we see here is that in the context of the One Health concept, vector-borne diseases such as vesicular stomatitis virus can have a severe impact of the health of animals, but it can also affect humans by reducing milk and meat production, which directly impacts food security. So what I show you here was just a little overview of how the research we do in the lab can actually help us to understand that influence of vectors, the biology of the vectors, and the transmission dynamics of specific viruses with very complex epidemiology. So for the last part of this seminar, I want to show you an example of how new technologies can be used as a tool to conduct viral surveillance. But these technologies has also allowed us to discover new viruses that can actually have the potential for biocontrol of pest and plant disease. So next generation sequencing is a novel technology that allows us to rapidly sequence millions or even billions of DNA or RNA fragments simultaneously. This technology can be used to identify viruses by comparing the sequences to existing databases. So we find matches to viruses that we know, but can also allow us to discover viruses by identifying sequences that they don't match to any known sequences. So we take those unknown sequences, we try to predict if the motifs of the protein are similar to the virus families we know. And if we have certain certainty, we can call those sequences as new viral species. So for this last part, I am just going to show you an overview of my current research here in UT, where I'm using RNA-seq in aphids. And just to give you a context, aphids are small sap-sucking insects. They're not as small as the midges that we talked before, but they're still pretty small. And aphids have a very high reproductive potential. They can adopt seasonal parthenogenic or asexual reproduction, in which you see the mom aphids giving birth to live clones. But those moms can also alternate to sexual reproduction, in which they lay eggs. So we are going to see female and male aphids. But this sexual reproduction is very limited to a specific seasonality. So you see them maybe at the end of the fall, and most of the life cycle of aphids, they are just reproducing asexually. Aphids, when they reproduce asexually, they can produce a lot of progeny in a very small time. So they become a serious agricultural pest because they can cause rapid damage to plants, but they are also remarkable vectors for plant viruses. So they can transmit everything and anything to plants and to crops. And another great point of studying aphids is because they have a very close symbiotic relationship with bacteria and viruses. So they are this super great model to study insect-microbe interactions. So to build functional resources for this project, I collect aphids infesting different host plants in agricultural and forest landscapes across East Tennessee. I target my collections on different host plants just to ensure aphid genetic diversity. So what you see here in the field is that I take those plants infected with aphids, I place the aphids in petri dishes with a coating of the leaf plant that I found in the field, and then I take them to the lab. In the lab, I start asexual colonies in lab-grown plants which are virus-free. And then I use those adult aphids that came from the field to perform RNA sequencing that I can later use to identify viruses. In addition to the science of this, this project has also allowed me to perform outreach activities in farming communities where I collect the aphids. So with these outreach activities, I have the opportunity to talk to farmers about the agricultural practices, listen to the perception and the impact of insect pests, but I can also provide some resources on insect identification and transmission to plant viruses by insect vectors. So going back to our science, the sequencing approach we're currently using has allowed us to find the composition of viruses occurring in wild aphid species. And we've found numerous plant viruses, so viruses that are infecting the plants and are potentially transmitted by aphids. But I have particularly focused in studying insect-specific virus that, if you remember, as I mentioned in the introduction, they're only viruses that infect only insects. They cannot infect plants and they cannot infect animals or humans. So what you see here in this chart is a heat map showing the relative abundance of hits to different viral families, taking those sequences and see which of the sequences, the percentage that match to virus families that we already know. So the virus families are here on the left, and the different aphid species are represented by these cartoons on top. The color represents a gradient of the number of reads aligning to those virus databases, being the reddish ones, the ones with the largest number of hits, which in this case was for the families Flaviviridae and Iflaviridae. So these two families are very interesting for me because they contain symbiotic virus with great potential to use for biocontrol. So talking a little bit about those two virus families, when we talk about the virus Flaviviridae, we see in RNA-seq dataset that they match mainly with a virus called MEV1. MEV1 was described in 2016, but it was described infecting only one species of tomato aphids. What we have found with this survey of field aphids across East Tennessee is that MEV1 can infect 3 other species of aphids commonly found in our local crops. And personally, this virus is very attractive to study because members of the family Flaviviridae present with a phenomenon called superinfection exclusion, in which the capacity of an aphid cell to infect with one virus, in this case MEV1, becomes resistant to further infection with a secondary virus, which can be the case of plant viruses, for example, potato leaf rot virus, which is transmitted by aphids. So what we're currently doing in the lab is trying to isolate this virus in cell culture to be able to have many copies of this virus so we can fully understand its potential as biocontrol. And I'm really hopeful that we can actually deploy this to control some of the disease that aphids transmit to potato and tomato plants. So the second family that we found in our sequencing data set and that had really high prevalence of matches was Iflaviridae. So Iflaviruses that we discover in our RNA-seq dataset correspond to multiple new species of viruses in aphids. So this virus species has never been described, and we found several species in our dataset. We found that these viruses persisted in the lab colonies that we maintain in the lab, and they are transmitted vertically from the mothers to the offspring. We have done an extensive work trying to find the transmission mechanisms, and we, without doubt, know that Iflaviruses are widespread heritable symbionts in aphids. So they persist in aphid populations. They're transmitted from mother twins to babies, and they just persist in the lines over and over again. We still don't know exactly the effects of these Iflaviruses in the host, but they seem to be persisting in the aphids with no consequences to their health. So aphids infected with Iflaviruses behave and reproduce exactly the same as non-infected aphids. From the biocontrol perspective, Iflaviruses are attractive because they can be used as virus-induced gene silencing vectors, which include the use of modified viruses, so taking these Iflaviruses, modify them in the lab in a way that they can alter the expression of target insect genes So for example, we take an inflavirus, we modify it in the lab in a way that the vector, for example, is going to reproduce less, it's going to have less reproductive potential. So by silencing these genes, they can feed less or reproduce less in certain plants, or even offer cross protection against plant pathogens. So we can deploy these viruses either as pest control or as a transmission blocking initiative for plant viruses. And because these viruses, these inflaviruses, are highly heritable, remember they pass from mothers to babies. Once the genetic virus is inserted in the desired population, we are expected to persist and be maintained in the pest species. So maintaining this control method can be very efficient. So what we see here, just to conclude this last portion of the talk, is that the new sequencing technologies are a great tool to conduct genomic surveillance. This genomic surveillance can allow us to monitor viral pathogens or emerging disease in insect population, but we can also track in real time how the emergence of a new infection or even follow the virus evolution during outbreaks. But outside an outbreak situation, so in our day-to-day lives, we can use these sequencing technologies to discover either new viruses that can be deployed as biocontrol mechanisms, or even can interact with other pathogens to reduce their transmission, or can interact directly with the insect health and act as biopesticide. So as I show you today, the research we do here at the university can have very big implications for animal, plant, and even human health. So the results I presented you today show that our research can not only contribute to the understanding of pathogen transmission by looking at the characteristics that make some of those insect species very good vectors, but it can also help us to understand pathogen emergence by exploring different mechanisms in which the viruses can persist. Lastly, we know that we can conduct pathogen surveillance using these new sequencing approaches, and we can use it in our day-to-day life here in the lab. But in the same way, some of the data deriving from these sequencing approaches can help us to find alternative tools to control insect pests and the viruses they transmit. So when we look at the overall linking of humans, animals, plants, and the environment, which involves this One Health approach, we see that this interlink in all the possibilities can allow us to address the full spectrum of disease control, prevention, and monitoring. So I do believe that promote a collaboration across disciplines it's very important to address the current and emerging health challenges we're seeing with the changing conditions. It can help us to promote food security and ensure a healthy balance in our ecosystems. So with that, I want to thank everybody for your time. Thank you for listening to this talk, and I'll be more than happy to take any questions.
Thank you so much. Wow, that was an awesome talk. Really, really appreciate that. Yes, we have time for some questions. I will say that aphids have been a thorn in my side for the past couple of years, dealing with my squash plants. They have taken over.
They love squash, and once they colonize one plant, they just spread. They spread so fast that there is really not a good method to catch up with transmission. With the reproduction. So once you see one, there is going to be a couple ones, and there's just going to be 100, 1,000, and you kill one, and they produce like 10 times more. So it's they're not easy.
No, and I've watched them start at one end of my garden and go all the way through it in just a matter of a couple of days.
That is absolutely true. So what my husband and I, we also own a organic farm. And what we do in our fields is we use decoy plants. So we use plants that are really tasty for those aphids, and we place them in different sites of the field just to try to lure them to the plants that we don't care. So the plants that we use for production suffer less than the plants that we don't care about. So that's it's not great. You're not going to get rid of it, but it allows us to coexist and still have some pretty good yields in our crops.
Oh, nice. I'm going to try that. I think there's a question in the chat from Dr. Cunningham, who's asking, do you have concern about the ability to get additional funding for this research in the current political climate?
I mean, of course, so we are all scientists. We're concerned about our research programs. It's changing. We have less and less funds, and it's becoming more and more competitive. In my case, I do hope that direct link that I have with agriculture can help me to get some funding. Like trying to find new control mechanisms, trying to understand disease transmission. I'm just hopeful, but I know it's going to be challenging.
Oh, and then there's a question, what are the tasty plants that attract aphids?
Well, it will depend on the aphid species, right? There are species that are very specific about the host, the plant they like, but there are other species that they like everything. So, for example, when we have a garden with mysus that are very prolific pests, so I found that they will like more cabbage plants than tomato plants, for example. So if I have a crop with tomatoes and peppers, I plant some cabbage around, and that really help us to lure those aphids to the other ones. In the case of grapes, we have grapevines. We put some other plants that are like little herbs around the garden, and that will lure them away from the vines. But it will depend on your specific situation. So if people are interested, I'll be more than happy to see the pictures of the aphids and try to suggest something. So my email was on the bottom of my presentation. Just feel free to email me and I will try my best to help.
And I think there was a question.
Yeah,
Go ahead.
Mohammed had his hand raised.
Oh, yes. Hi.
Yeah, go ahead.
Hi.
Yeah, thank you, Paula, for this nice talk and presentation. I'm wondering if I'm catch up. Have you tried those virus culture in the lab? Which lines you grow those viruses, novel viruses? Can you please elaborate about this, please?
Yes, absolutely. That's a very interesting question. So you usually want to isolate viruses in the cell lines of the insect that you find it, right? And that is true for the case of the culicoides virus. We have insect cell lines that are culicoides cells, so vesicular stomatitis virus grows really, really well. In the case of those insect-specific viruses, we do not have an aphid cell line. So I'm trying my best to use other insect cell lines that will allow me to grow these viruses. And that approach is family dependent. So for example, for the flaviviruses, I'm trying to use mosquito cell lines because mosquito cell lines, they're very good vectors of flaviviruses. So dengue, yellow fever, those are flaviviruses. So I'm trying to use this mosquito cell line just to see if by having the same virus family is going to allow me to grow them. For the case of the viruses, they're very, very specific. So I'm trying to use evolutionary closely related insects like leaf hoppers, for example, and see if maybe, like I force the cells to uptake the virus, they can replicate it inside.
Okay, thank you.
I had myself muted. We have time for a couple more questions if anybody has any additional questions. I find it really fascinating, just how you might be able to use it as like a biological control.
Yeah, that is kind of like the cherry on top of the project. It as I showed you, just this overview of working with the farming communities and finding new species of virus, so this virus discovery, but having this biocontrol potential was just the cherry on top for the project. It really complete the whole story of how science, can help us to get something that is affecting the farming communities, trying to do our natural and our evolutionary research in the lab, but also having a product that we can try to deploy to improve the situation that we see in the field.
Mhm. I work a lot with animal species, so in my mind, I'm going through how we could apply this to animals as well.
Well, there are some insect-specific viruses that they're trying to use for mosquitoes, for example. So for either human or animal disease, and they are calling these transmission blocking effects, in which you put the virus in the mosquito and they cannot transmit the disease, or you put a portion of the virus that it will make the animal sick, for example, but it's enough to build immunity, so it works as a vaccine. So those initiatives, they're right now, there's groups trying to understand how it's happening and try to use them for dengue and for malaria, for example. So we're just hoping that they can get something, because it looks really exciting, like if you can vaccinate the vector population, so either animals or humans don't get sick, that it will be very interesting to see.
Yeah, absolutely. That would be, wow, there would be so many applications. That'd be amazing.
Yeah, so we're hopeful that we can get some really good funding and we can continue our research.
I saw a comment in the chat, said congratulations on this important work. It's great that you found a positive use for the pesky midges. A good reminder that even disturbing things can have positive roles. They said, if I hate to ask this, but kind of what you were just saying, do you have any concerns about your ability to get some additional funding for the research in this current political climate? They said, I hate to ask.
Yeah, I mean, as I mentioned, at the beginning of the question session, I think we are all concerned. It doesn't matter what you work on. We're all trying our best. But, I just have to be hopeful that, it's going to get better. We always find ways to make things work. And, we just have to keep doing what we do best,
I think
Absolutely.
because of the importance of food, we have such a, food insecurity issues, nutrition insecurity, that your work is going, in that realm is like, should be sustainable and fundable, because of that, because there's such great need. And, if we have most of agricultural crops or being destroyed by insects, that's, takes away from productivity. And, the more that we can produce on the land we have, the healthier crops and have less loss, the better, right?
Yeah, definitely.
Yeah. And I feel like Tennessee's been pretty supportive of agriculture.
I do think, I mean, it's kind of the main economic motors of the state, right? We're really good in agriculture. We not only crops but also livestock. So I do believe that, at least from the state perspective, there is still some support.
Mhm.
And it's just being hopeful that hold on for the next few years, and it will get better. We will go back to what makes science in this country great.
Mhm. Absolutely. All right. Anything else? I have one last question, I guess. When, and maybe you haven't come across this yet, but if you have had a virus, because I have come across this in the animal realm, a virus that doesn't really fit anywhere. Have you come across that yet? And the challenge of trying to figure out, is it significant? Is it incidental? Yeah.
Yes, we actually have. So we have either viruses that don't really match to anything but look like something. So we're trying, for the computational perspective, we're just trying to find the models in those proteins and to see if those motifs can give us certain certainty that we're in a virus family, for example, right? But we also found viruses that we only have partial sequences. So it's like, it looks like something like a virus, but it doesn't really, we don't find anything else. And we're like, and we keep finding this partial sequence of one or two proteins. And what we found was actually that these viruses were inserted in the aphid genome. So they were endogenized. And now they belong to the insect genome, but they keep expressing only one or two proteins. So you really don't have a new virus. You just have viruses that belong now to the aphid. And that was a beautiful story that, I couldn't really fit for today's talk, but it seems that some of those viruses are related to make aphids produce wings so they can disperse. So yeah, we have found both.
Oh, interesting. Wow. Yeah, I have something very similar, and it's something that I came across years ago, and I'm wishing that I'm like, how could we have such better techniques now? Could I possibly do a better job at identifying it now? And I'm not seeing it again. So yeah, that's awesome. And we do have another question. Do pesticides affect the evolution of the viruses you're studying?
I believe so. I'm not 100% sure because I haven't really looked into it, but I do believe that either by the selection pressure that the pesticide does, so it will select certain specific genotypes in the pest, for example, that will lead to virus evolution. But also there are some virus families that seem to either affect certain resistance to the pest, to these detoxification effects when they're exposed to pesticides. So we'll see those viruses are going to become more prevalent in the pest because they're allowing them to survive. So I believe it's happening. I just really haven't studied it.
It looks like we're kind of coming up on the end of the hour. So if there are no more questions, all right.
Well, thank you for the invitation. It was a pleasure talking to you.
Thank you so much.
Yes,
It was
thank you.
really an awesome presentation, and I learned a lot, and yeah, now I know who to contact, especially for my aphid issues.
Absolutely. I love what I do, so I'm more than happy when people email me and ask questions. I'm just really happy that I can help and I can use my science and what I know.
Yeah. Thank you so much.
Well,
And I might also contact you regarding some virus ID issues too.
Oh, absolutely. I would love to talk. Yes. So thank you again.
All right. Bye-bye.